Okay, good morning everyone, good morning. Okay, that's right for a minute. Good morning everyone, please confirm, am I audible? Good morning, good morning. Okay. Okay, good morning, perfect. Good morning. Okay, so friends, from today onwards, we will be starting this particular revision course. Okay, so I'm requesting every one of you first just listen to me very clearly. Okay, so in this particular revision course, we will be discussing about the minimum 60 to 65 topics compulsory. Okay, see friends, I have analyzed carefully the 2022 February paper, followed by the 2022 September paper, then followed by the 2020 November paper. So friends, based on the thorough analysis, see, I have listed out all these topics. See, and we are going to discuss this also. When I analyzed the three question papers, 77 to 78% of all the questions that has been directly asked from our PPT itself. So if you revise the PPT once and if you read the Pathfinder once, it's more than sufficient to crack the exam in simple attempt or in a first attempt. So I know that many repeater students are also studying, so I'm requesting all of you kindly make use of this particular course completely. Okay, so friends, now the most important, you have to remember what are the various topics that we are going to study. Okay, see, and first let me tell you the plan. See, every single day the class will be from, everyone please look at this, every single day the class will be 5:45 a.m. to 6:45 a.m. in the YouTube live, then followed by the class will be continued 11:30 to 12:30 p.m. in the app. You have to attend the next class continuously only from the app. Okay, so whatever the topic that I am going to discuss in the morning YouTube class as well as in the app, we are going to discuss this, we are going to do the study hour on the same topic every day at the 7 to 8:30 p.m. So what I will be doing in this particular study hour, suppose example today we are going to discuss in the two classes two important hormones. So in the study hour, what exactly we are doing is we will be discussing, we will be doing the self study. So you will be also studying the topic and I will be also studying the topic, and I will do the instant doubt solving on that particular topic only. After the completion of study hour, we will discuss the PYQ questions specifically asked from only this particular topic. Okay, so friends, the only request from my side to all of you is that just be consistent for the next 45 days. Okay, if you are really consistent for the next 45 days, friends, I'm not giving the 100% assurance, I'm giving the 85% of assurance that more than 110 to 120 marks you can expect from the topics that what we are going to study. Okay, so I am requesting every student maintain three things. One is consistency, second most important is the interest, and be patient throughout the class, because I will explain every concept from very basic level to the very advanced level. By that, if you do the one time revision, that is more than sufficient to remember as well as the practice. Okay, so now I have listed out different chapters as well as the different units. Okay, now friends, if you look into this plant physiology, let's consider the September 2022 paper. We got plant hormones. How many questions we got from the plant hormones? We got 6 marks only from the plant hormones, and that too surprisingly friends, we got the 6 marks only from one hormone, that is the gibberellic acid. Part B also we got the question from gibberellic acid only. Part C also we got the question from the gibberellic acid only, surprisingly, but it never happens all the time, but I am just saying. Then we got photosynthesis only 2 mark, and we got the question from the secondary metabolites 4 mark. Sensory photobiology we got 4 mark, and I forgot to keep another topic, there is the transport in plants, which we got the 4 mark question. So today we will be starting about the unit 6. Okay, so and friends, I don't know whether you have seen the question paper. Good morning everyone. I really don't know whether you have seen the question paper correctly or not, September 2022. If you see the question paper, maximum number of marks that was appeared from the chapter of ecology. If you please count the marks, we got 32, I think 34 marks from this particular chapter of ecology only. And you know, 6 4-marker questions were appeared, as well as 5 2-marker questions also appeared from the ecology. Most of the students will start, most of the students will neglect the chapter of ecology because it's a very rigid chapter, and it's a kind of a dry chapter where you have to remember many things. And surprisingly friends, whatever the questions asked from this particular chapter of ecology are very direct. Out of 6 part C questions, only 2 part C questions are little tough questions, and remaining 4 part C questions are very straightforward, even directly from the PPT. I will show you when we are discussing. So after the unit 10, we will be doing the unit 3, then we will be doing the unit 2, and we will be doing unit 1, then followed by the unit 5, then followed by the unit 7, unit 4, then unit 8, unit 11, then followed by the unit 9 and 13. Okay, so once again I am requesting all of you friends, just be consistent throughout your preparation. That is the only thing I am expecting from everyone. Okay, so and especially the people who are repeaters, I am requesting every one of you friends, please attend the study hour compulsory. The major reason of keeping study hour is most of the people are unable to study on their own, and most of the people are not understanding what to study or what not to study, and most importantly how to study. Most importantly, how much extent to study. Many people will say that in the reference book there is a lot of information, and the Pathfinder also there is a lot of information. So I will also sit with the Pathfinder in the evening class along with the PPT, and I will tell you what exactly you people has to read and how much time to read. By that, it will become a good practice for all of you. Okay, and friends, requesting every one of you last and final time, be patient throughout the class, then only you can learn much. Okay, now let's start. Plant hormones, right? So friends, in this particular concept of the plant hormones, as I said that we got the 6 marks, especially from the one single hormone, that is what friends, gibberellic acid. But I am not at all saying that every time we will get the question only from the gibberellic acid. So that is why you have to study each and every hormone compulsory. Okay, so friends, whenever we are reading the hormones, remember my words, what kind of questions that can be expected from this particular hormone. Suppose example, let's take the 2022 February paper. What kind of question we got? We got the question from the matching. You know it very well, right? But if you look into the question paper of September 2022, the part B question appeared from where, friends? The part B question appeared from the functions, and the most importantly from where the part C question was appeared, the part C question were appeared from the process of signaling, right? So matching, functions, signaling. Otherwise they can also ask the question from the precursors involved over there, means which hormone contains what kind of a precursor. Even the question will appear from this particular concept also. So while studying the hormone, in a simple words, you have to study from A to Z. Once you study from A to Z, you can answer any kind of a question. The point is here, 99.99% of the time, the question will be direct question from the hormones. And most importantly, you will be getting the question for sure. Okay. Now let's start. So friends, how can we define the word hormones? Everyone define? Please follow me very correctly. How can we define the word hormones? Hormones are said to be the chemical messengers. Be patient. So why we are calling hormones are the chemical messengers? Because friends, they are carrying the information from one area to the another area. So that's the reason we are calling it as the chemical messengers. And most importantly, does the hormones having any kind of a nutritional value, friends? There's no. We always call hormones are non-nutritious in nature because they do not have any kind of a nutritional value. Most important, friends, thus, everyone look into the point. The site of production and the site of action is entirely different. What do you mean by that, friends? We all people know that hormone produces at one particular area, but the hormone is going to show their action in the other area. Is it, friends? What do you mean by that? Which means, suppose example, let's take a hormone auxin, where the hormone auxin is going to be produced. Hormone auxin produced in the shoot, but where the hormone auxin is going to show its action in the root. So production is different, action is entirely different. That's why we can say site of production and site of action is entirely different. But friends, everyone remember my words. What most of the people will think that if the hormones want to show the better action, people think that hormones should be released in a very high concentration. But the point every student has to remember for the exam, if the hormone releasing in the higher concentration, the activity will become extremely lower. So what's the point you have to remember? If the hormone want to show the maximum activity, it should be released always in a very low concentration. In a simple words, low the concentration, high the activity. Suppose high the concentration, low the activity. See, not only the plant hormone I am talking about, even if you consider the animal hormones also, even the hormones present in our body also, any hormone if it wants to release show action in our body, it has to release in the very low concentration. And very unique point, you know very well, if whether it is a plant hormone or whether it is the animal hormone, doesn't matter. If a hormone want to show the action, compulsory the hormone has to attach to what, friends? Receptors. Yes or no? Without attaching to the receptor, no hormone can show their action. Why? Because if the hormone want to show the action, it has to come and attach us to its specific binding site, which is present on the plasma membrane, which is present on the cell wall, which is present on the nuclear membrane, or which is present within the nucleus, or which is present within the cytoplasm. In a simple words, hormone want to show their action, it has to compulsory attach to what, friends? Receptors. So let me quickly summarize this particular slide, friends. Hormones are the chemical messengers because they are involved in transferring the information or transferring the message from one part of the plant body to the another part of the plant body. And we are calling plant hormones are non-nutritious in nature because they do not contain any kind of a nutritional value, right? And most importantly, hormones produced at one particular part and it is going to show their action on the other part. Please don't confuse with that. Another important point, friends, if the plant hormone want to show their action, it has to be released in a very low concentration. Most unique point, any hormone want to show their action, it has to be released in a very low concentration and it has to attach to the receptor molecules, right? Okay, now friends, what are the various type of hormones that we people has to study majorly? Okay, majorly we have to study the auxin, gibberellic acid, abscisic acid, ethylene, jasmonic acid. Friends, please remember, I want you people to study even brassinosteroids also. You know, brassinosteroids, I will tell you what exactly to be read in the Pathfinder, as well as I will tell what exactly to read about the jasmonic acid also in the Pathfinder. Very small information is there. I'll tell exactly what to read. I want you to focus on only that particular information. Okay. Now, what are the major 5 hormones that we have to focus? Auxin, abscisic acid, cytokinin, gibberellic acid and ethylene. So friends, first we have to understand from where these hormones are derived from. Okay, the first important, I have written here 5 major hormones. The point is very clear. How did I classify these 5 hormones? Based on what? Very important point. Friends, all the hormones present in the plant body broadly categorized into 2 types. Okay, based on what we broadly categorized them into 2 types? We categorized them into 2 types, whether they are promoting the growth or they are inhibiting the growth. So in a simple words, we categorized all the hormones into 2 major types. What are those 2 major types, friends? One we can call it as the promoters, another one we can call it as the inhibitors. Okay, if a hormone promote the growth of a plant, we can call growth promoter. If a hormone inhibit the growth of a plant, we will be calling it as the growth inhibitor. Everyone follow my words correctly. What are the most important growth promoter hormones present in the plant body? Auxin. Auxin is a very important growth promoter. And friends, cytokinin, which is an important growth promoter. And friends, another most important one is what? Gibberellic acid. These 3 are the major growth promoters. And we started studying from the 11th standard onwards. What is the most important growth inhibitor present in the plant body, friends? Most important inhibitor, which is the abscisic acid. We all people know that very well. Okay, apart from the abscisic acid, there is one more hormone is left. What is that hormone is left? Which is the ethylene. Correct na? Ethylene is the another hormone. Now the point is very clear. What is the role of a ethylene? Does ethylene can act as a growth promoter or does ethylene can act as a growth inhibitor? Everyone please keep it in your mind. Ethylene, depending on the conditions, depending on a particular condition, it will be acting as a growth promoter, and depending on the requirement or condition, it can act as a inhibitor also. So keep it in your mind. Ethylene is a kind of exceptional hormone which can act as a promoter also, which can act as a inhibitor also, depending on the condition. Whereas abscisic acid can act as purely inhibitor, whereas auxin, gibberellic acid, cytokinin act as a purely growth promoters only. Don't get confused with this particular point. And friends, now the most important I want you people to remember is that the derivatives means what are the precursors that are involved in the synthesis of these particular hormones. From here also we can expect the question in the part C directly, especially in the matching questions. Which hormone is derived from which precursor? Okay, you have to remember compulsory, friends. Okay, auxin. Auxin is basically derived from what derivatives? I hope you know, we are going to study it. We called most important auxin is the indole acetic acid. Correct now, friends? Now, this auxin is actually derived from what, friends? Indole derivatives. Okay, auxin is derived from what? Indole derivatives. That's why we can call it indole acetic acid, we can call it indole butyric acid. So keep it in your mind, auxin is the derivative of indole, right? And what about the abscisic acid, friends? And what about the cytokinin as well as gibberellic acid as well as the ethylene? You don't need to think too much about the ethylene. Ethylene is what kind of a plant hormone, friends? We all people know that ethylene is the only hormone which is a gaseous in nature in the plant body. Correct, na? Then gibberellic acid, the part B question which were asked in the September 2022 paper, which is a straightforward question. The gibberellic acid is the derivative of what? The gibberellic acid is the derivative of what? A terpene. They have asked it belongs to which terpene? Whether it is the monoterpene, whether it is the diterpene, they have asked. Of course, we are going to discuss that. Gibberellic acid is the diterpene which is having the 20 carbon containing compounds. We will discuss that. Now, abscisic acid. It is derived from what, friends? Everyone remember that? Abscisic acid majorly derived from, friends, carotenoids. Abscisic acid is derived from what, friends? Carotenoids. You know, right? Friends, carotenoids also basically comes under the terpenes only, but we never say that abscisic acid is directly synthesized from the terpenes. What we can say that one of the terpene derivative is the carotenoid. So from the carotenoid, this particular abscisic acid got derived. Okay. And what about this particular hormone cytokinin? Cytokinin exclusively derived from the purines. Exclusively derived from what, friends? Purines. And especially which purine? So there is a adenine also, there is a guanine also. Friends, cytokinin exclusively derived from the adenine. That's why we can call cytokinin as aminopurine. We will discuss that. So remember everyone, for the matching type of questions, auxin is a indole derivative, abscisic acid is a carotenoid, cytokinin is the purine derivative, gibberellic acid is the terpene, ethylene is the major and simple gaseous hormone. And we categorize all the hormones into two major classes based on whether they are promoting the growth or whether they are inhibiting the growth, which we can call growth promoter and growth inhibitor. So now we are starting with the first and most important hormone, friends, that is the auxin. Okay, now the question is here, sir, why we are starting only from the auxin? Okay, we will study from most important hormone to the next, because why we are discussing only, anyway we have to discuss every hormone, but when compared to all other hormones, the hormone auxin was asked in the exam multiple times. Every hormone was asked many times, but when compared to all, auxin was asked multiple times. So that's why we are starting with the auxin. So once again, I am telling you what we have to study whenever we are studying this particular hormone. Any hormone, okay? First, try to understand the discovery. Okay, now the question is here, sir, do we really get the question from the discovery? No, we don't get it. But we have to study the discovery in order to understand the hormone. Next, we have to understand what kind of precursor is involved in the auxin biosynthesis. Most important, the biosynthetic pathway we have to study. Right. After studying the biosynthetic pathway, we have to study about the transport, how the auxin is actually transporting in the plant body, from where to where it is transporting. After studying the transport, we have to study the auxin from, we have to study about the functions. Finally, after studying the function, every student has to remember the signaling. Everyone has to study the signaling. Whether you believe it or not, the question never go beyond whatever we are studying. Okay, so beyond this level, there is no other way of asking the question only. How many, irrespective of how many number of question papers you may see, the questions only appear from this. See, whenever you are reading, friends, you have to read always towards the exam point of view. You should not read randomly, or you should not read A to Z, which never ask in the exam. Since we are restricted to only one particular competitive exam, we should always read towards the competitive exam only. Okay. So friends, now let's start studying about the discovery first. Okay, and once again I am telling to all of you friends that from the discovery you don't get any kind of a question at all. Okay, but in order to understand a hormone, it is very important every student has to study the discovery. Okay, now let me explain you the discovery. In the year 1880, okay, I will explain in 5 minutes. Every student pay attention very correctly. In the year 1880, Charles Darwin and his son Francis Darwin, Charles Darwin and his son Francis Darwin, they started doing the experiment. Where they started doing the experiment? They started doing the experiment on the coleoptile. What they are doing? They started doing the experiment on the coleoptile. Now the question is very clear, sir, what is coleoptile? Where it is present in the plant body? I have never seen a coleoptile. So every student look into this. Suppose this is a shoot. Friends, suppose this is a shoot. Now remember, this is called protective covering. What do you call? The sheath or protective covering. Remember my words correctly. Protective sheath or protective covering of the shoot, which we can call it as coleoptile. Okay, so Darwin and his son started doing the experiments on the coleoptile. Whenever they are doing the experiment on the coleoptile, friends, you know what they have done? They have exposed the coleoptile to the unilateral sunlight. My friends, everyone understand what do you mean by the word unilateral sunlight? Unilateral sunlight means the sunlight is coming only in one direction. Suppose I am standing here, suppose the sunlight is directly falling on, light is falling on my face. So what I called unilateral direction. So they exposed the coleoptile to the unilateral direction. Then what they observed? They observed the coleoptile started bending like this. The coleoptile started bending like this towards the light. Now friends, everyone remember my words very correctly. You know what Darwin and his son concluded? What the Darwin and his son concluded? Why the coleoptile got bent? Darwin and his son concluded that there is a chemical. There is a chemical which is present in the tip of the coleoptile. That chemical always moving from where to where? The chemical is always moving from lighter region to the darker region. So you know, right? So when we expose it to the sunlight, there is a lot of light. So because of that light, whatever the chemical that is exactly present in this particular tip, it starts moving towards the darker region. Whenever it starts moving towards the darker region, what happens, you know? The cell get elongated. Understand my point very correctly. The cell get elongated. Because of the elongation of the cell, the coleoptile is exactly like this. So because of the elongation of the cell, what is happening, friends? The coleoptile starts bending. That's a simple concept. But unfortunately, the Darwin and his son did not get any kind of a credit for this particular simple experiment. But later, there are many scientists who does the experiment, friends. But an intelligent scientist named as Francis Siswent. Okay, Francis Siswent. What the Went does, everyone please understand what exactly I am saying. There is a scientist called as Francis Siswent. What the Went does, all the students follow me. Same, he also taken the coleoptile. Okay, friends, you know from where he has taken the coleoptile? He has taken the coleoptile from the Avena grass seedlings. What are Avena grass? Common oat plant. Okay, follow my words. So from the Avena grass seedlings, nothing but common oat plant, he has taken the coleoptile. The moment he has taken the coleoptile, you know what happened? Same. He exposed to the, understand my words, he exposed to the unilateral sunlight. My friends, what happened? Then obviously it starts bending immediately. Now he got a good idea. So why the coleoptile is bending? When they exposed it to the unilateral sunlight, he got a valid doubt. Now this time what he has done, you know, he has taken the coleoptile. All the students follow me. He cut the coleoptile. Okay, he cut the coleoptile. Where he cut? Not everywhere. He cut the coleoptile. I will explain twice. Focus. He cut the coleoptile only on the top here. Exactly. He cut the tip. What, friends? He cut the tip. Whatever the tip that he cut, you know what he does? He kept in the agar solution. Follow my words correctly. Where he kept in the agar solution. Now every student has to ask me, sir, why only in the agar solution? He kept anywhere, right? Because if you keep something in the agar solution, always it diffuses it. It will always take inside, right? So that's why he kept in the agar solution. Now everyone follow me. Now without cap, how does it look like? Without cap, it looks like this. You know what do we call it as without cap? We can call it as decapitated coleoptile. My friends, what do we call it? Decapitated coleoptile without cap. Now you know what has done. Without cap, he exposed to the unilateral sunlight once again. And friends, surprisingly, it is not at all bending. It is not at all bending. Now you know what he has done. Once again, whatever the tip that he kept in the agar solution, he kept the tip back onto the coleoptile. He kept the tip back onto the decapitated part and then he exposed it to the sunlight. Now this time you know what happens? This time it starts bending. So what is the conclusion we have to understand? We have to understand a conclusion that yes, there is a chemical which is involved in the bending of the coleoptile. There is no doubt about it. But the point is that where that chemical is present, that chemical is exclusively present only in the tip of the coleoptile only. Remember once again what the Francis Siswent does. He has taken the coleoptile. What is the coleoptile? Protective sheath, protective covering of a shoot from the Avena grass seedlings, nothing but common oat plant. And after taking that, he exposed it to the unilateral sunlight. Then what happened? It starts bending. Now to understand why it is bending, what he has done, he cut the tip. Whatever the tip that he cut, he kept in the agar solution for a particular period of time. Now without cap, what do we call it as, friends? Decapitated part. This decapitated part, he exposed it to the unilateral sunlight. Now it's not at all bending. Now what he has done again, whatever the tip that he kept in the agar solution, he kept back on the decapitated part. Then he exposed again. Then it starts bending. Now he got a complete idea. Yes, there is a chemical present in the tip of the coleoptile, moving from which region to which region? Lighter region to the darker region. Whenever it is moving from lighter region to the darker region, it is causing cell to get elongated. Remember my words, it is causing all the time cell elongation. Friends, it is causing the cell elongation. That is the reason the coleoptile starts bending. This test called as Avena coleoptile curvature test. We can say that this is the major confirmatory test to identify the presence of auxin. As I said that there is a chemical is present in the coleoptile. Siswent is the scientist who isolated that chemical. What, friends? Who isolated that chemical from the tip and given the name auxin. You know what exactly the word auxin means? Auxin means to grow. Auxin means to grow, which is helpful in the growth. How it is helpful in the growth? It is helpful in the growth by the process of elongation. Never ever get confused. So Siswent is the first scientist who has given the name auxin, which means to grow. Later, there is another scientist called as Kogel. There is another scientist called as Kogel. You know what he has done, friends? He has isolated a very low concentration of auxin from the human urine. Okay, from the human urine. every student has to remember a unique point is that till today we really did not understand what is the exact role of a auxin in the human urine, and we did not found a very high concentration of auxin also, friends. We found very low concentration of auxin, but we really don't know what exact role of that low concentration of auxin also. Still, there are many studies has been conducted, but we did not come to a valid conclusion what is the exact role. So friends, this will be the simple conclusion of what, friends? This will be the simple conclusion of the discovery of auxin. Why I explained the discovery is just to make you understand how the auxin was discovered. If you can remember for the exam point of view, otherwise there is no problem at all. Okay, now from now onwards, whatever we are going to study, friends, everything will be most important. Okay, so please be patient throughout the class, understand the concepts very correctly, by that you can revise very faster. Okay, friends, what are the types of auxin? See, there are many types of auxins are present in the plant body, but depending on how they got derived, we categorize them into different types. So the first one we can call the natural auxins. Every student follow me. How can I define the word natural auxin? Name itself indicating, right? The hormone, the auxin which is derived naturally in the plant body. That is what exactly we can call natural auxin. What are the natural auxin examples? IAA, IBA, indole acetic acid and indole butyric acid. And one more point every student has to remember for the exam. It doesn't matter how many number of natural auxins are present in the plant body, keep it in your mind, but every time auxin mostly exhibit in the IAA form. I can use a word that the predominant form, remember my words, the predominant form or the dominant form of auxin in the plant body is always IAA form only. Never forget that point. Okay, natural auxins means which we have synthesized naturally in the plant body. And what about the synthetic auxins? The name itself indicating which are man-made, which are synthesized synthetically, correct? Now laboratory. Now friends, very important, you have to remember for the exam, what are the best examples of the synthetic auxin? This is also asked part B question, which is NPA, naphthyl thalamic acid. What is that? Naphthyl thalamic acid. The next one, 2,4 dichlorophenoxyacetic acid. What is that, friends? 2,4 dichlorophenoxyacetic acid. And what will be the another example? 2,4,5 trichlorophenoxyacetic acid. What is that? 2,4 dichlorophenoxyacetic acid, 2,4,5 trichlorophenoxyacetic acid. Very easy to remember, right? 2,4 are the carbon numbers where the chlorofunctional groups are present. Simple. 2,4,5 are the carbon numbers where the chlorofunctional groups are present. That's what. So very important point everyone has to remember, friends. These particular 2,4 dichlorophenoxyacetic acid and trichlorophenoxyacetic acid, they are all actually acting like a weedicides. Okay, which are involved in killing the weeds. Please don't get confused with this particular point at all. Now, don't confuse, friends. Now you understand what is natural auxin, which are synthesized naturally in the plant body. Synthetic auxins, which are synthesized, in the laboratory or man-made. Now, don't confuse with these two definitions. That is the free auxin and bound auxin. Don't confuse. Listen to me correctly, friends. A free auxin may be anything. Remember my words. A free auxin may be a natural auxin. A free auxin may be a synthetic auxin. It may be anything. Then why I have given the name free auxin? When I synthesize a auxin without using any organic solvent. Remember my words correctly. When I synthesize a auxin without using any organic solvent, then I will call that auxin as the free auxin. Means without there will be another method, but we don't use any organic solvent while we are synthesizing that. So you can call a natural auxin also a free auxin. You can call a synthetic auxin also a free auxin if we synthesized without using organic solvent. That is called free auxin. Simple. And which auxin? You will be calling it as the bound auxin. If I want to synthesize a auxin, if I am using an organic solvent, if I am using an organic solvent, any kind of a organic solvent, let's take an example of ethanol, chloroform, carbon tetrachloride, whatever it may be. If you use a organic solvent in the synthesis of a auxin, we call that auxin as the bound auxin. So once again friends, a bound auxin may be a natural auxin, a bound auxin may be a synthetic auxin. If you use organic solvent, it will become a bound auxin. So you know what is natural auxin, you know what is synthetic auxin, you know what is free auxin, you know what is bound auxin. Friends, there will be a very specialized type is there, which we call it as antioxins. What do we call it, friends? Antioxins. Name itself indicating now friends, which is acting opposite to the auxin. Yes or no? Antioxin. I have told you multiple times, even in the human also. One hormone, remember my words, one hormone stimulates the action of other hormone, one hormone inhibits the action of other hormone. You know very well, which means insulin always inhibits the action of glucagon, or vice versa. Glucagon inhibits the action of insulin. Similarly, if any hormone, follow my words correctly, if any hormone inhibit the action of auxin, we call that particular hormone as antioxin. So let's take an inhibitor. See, don't think that every time inhibitors only has to come and inhibit the hormonal action. It's nothing like that. One hormone can inhibit the action of other hormone also. So then what we can say, if any hormone blocking the action of that auxin hormone, then we can call it them also antioxins. I hope everyone understand that without having any kind of the confusion, right? Okay, so friends, I am teaching little slowly, by that you will understand every point while you are reading. That should be your final revision. You have to go immediately for the question solving. So make sure that you learn the concept very correctly. Okay, now where the hormone auxin get biosynthesized, very important point, as we are already studied that, and I said the production of the hormone, that is site of production, site of action, is entirely different. Friends, everyone remember my words. The moment you heard the name of a auxin, one point has to strike in your mind. The point that has to strike in your mind is that auxin is the rooting hormone. What hormone the auxin is? Rooting hormone. Now the point is here, when I use the word auxin is a rooting hormone, does the auxin is a synthesizer in the root? That is absolutely wrong, because hormone produced at one place and show their action at another place. So where the hormone auxin is produced, hormone auxin is always producing at the shoot. Where exactly at the shoot? Apical meristem of the shoot. Everyone remember, apical meristem of the shoot is the site where the auxin is getting produced or synthesized. From that particular region, auxin is traveling where? Auxin is traveling till the root. Till the root, auxin will travel and it will show their entire action only in the root, not anywhere else. Mostly, I am talking about 85% of the auxin hormonal action will be on the root only. That is the reason I have given a statement that auxin is a major rooting hormone. So, shoot apical meristem or the major sites of the synthesis of auxin. Don't confuse with my words. I can say that 90% of the auxin, friends, will be synthesized. What, friends? 90% of the auxin will be synthesized, in the apical meristem. What about the remaining 10%? Don't confuse. The remaining 10% of the auxin will be synthesized in all parts of the plant body. Don't confuse. All parts of the plant body, auxin will be present, but not in the higher concentration. Extremely lower concentration, auxin will be present in the all parts, very low. But major concentration of auxin is accumulated only in the shoot apical meristem. Now everyone remember. Auxin biosynthesis. Now I want every student follow me very correctly. Is there any necessity to remember the structures present in this particular auxin biosynthesis? No. Okay. Now, if you look into our 2022 September question paper, how many structure related questions we got? I don't know whether you remember or not. We got only one question which is structural related. That is also question from the glycolysis they have asked with respect to the phosphofructokinase. Apart from that, we did not get any kind of a structural based question. Anyway, that forward, that is the biochemistry that time we will discuss. But when it comes to the plant physiology chapter, you people no need to remember any kind of a structure, but there are one or two structures to be remembered. I will tell what you have to be remembered. Okay. Now we are studying the pathway. Now you can ask me, sir, what exactly we have to remember in the pathway? Everyone remember my words. You have to remember the schematic presentation. What do you have to remember? Schematic presentation, which means, listen my words, which is the starting precursor, which is the intermediate molecule, which is the ending molecule. What? Which is the starting precursor, which is the intermediate molecule, which is the ending molecule. Apart from this, you no need to read anything, no need to remember anything. Okay. Now, once again, the asked question. Which amino acid can act as a precursor for the biosynthesis of auxin? So, as we all people know that, which is the asked part B question, which is the tryptophan, which can be acting as the precursor molecule. Friends, how can you define the word precursor molecule? Precursor means starting compound, which is starting the synthesis. That is called precursor. Now everyone has to get a doubt. There are 20 amino acids are there. Why tryptophan only acting as the precursor? What's the reason? You have to ask yourself. Because tryptophan having what ring in the structure? It is having indole ring. We know very well, and we already studied that auxin is derived from which derivatives, friends? Auxin is derived from the indole derivatives. So which means a compound which is having the indole ring can basically act as the precursor molecule. Simple. That's why tryptophan can act as a precursor molecule for this particular auxin biosynthesis. Now all the students follow me. In the biosynthesis of auxin, there are many pathways are involved, many pathways. So there is a pathway called as indole 3-pyruvic acid pathway. Don't get confused, friends. There is a pathway called IAN pathway, indole acetonitrile. Don't confuse, indole acetonitrile pathway. There is another pathway called as IAM pathway, indole acetamido pathway. Indole acetamido pathway. There is another pathway called as TAM pathway, tryptamine pathway. Now the point is here, there are many pathways are there. Which pathway will be involved in the biosynthesis of auxin mostly in our plant body? That is the question. In the plant, always the IPA pathway will occur. Most predominant, once again understand, the most predominant pathway for the biosynthesis of auxin is the always IPA pathway only. Now you can ask me, sir, why not this? The question is here, they will also involved in the biosynthesis of auxin, but they will occur in the lower organisms. Let's take a bacteria. Right. So in the bacterial body, this IAN, IAM, TAM pathways occur. But when it comes to the plant, there is a IPA pathway, indole 3-pyruvic acid pathway. Very simple to remember. Understand my words correctly. What is the ultimate goal of a IPA pathway? Ultimate goal of the IPA pathway to synthesize auxin. From where? to synthesize the auxin from the tryptophan. That's all, nothing else. Synthesis of indole acetic acid from the tryptophan. That's all. That is the indole 3-pyruvic acid pathway. Now I will explain how. You people don't need to remember any structure. Understand the schematic presentation what I am going to explain to all of you. Look into this, friends. This is what? Tryptophan. Don't worry, it's very simple. Tryptophan is an amino acid, right? It is having what group? It is having carboxyl group. It is having amino group. All the students follow me. Now this tryptophan first treated by an important enzyme. What is the name of the enzyme? It is called as tryptophan transaminase enzyme. What enzyme? Tryptophan transaminase enzyme. You know, name itself indicating, right? Transaminase, which is transferring the amino group. Such a simple step it is. So because of the transfer of amino group, tryptophan is converting into what? Tryptophan is converting into IPA, indole 3-pyruvic acid. You can see it very easy, friends. There is a transfer of the amino group. Just because of the transfer of amino group, tryptophan converted into IPA pathway. Now you all has to understand why we have given the name IPA pathway. Why we have given the name IPA pathway? Because the stable product, the intermediate product produced is the IPA. Intermediate product is IPA. That's what name of the pathway also, IPA pathway. Simple. Now the indole 3-pyruvic acid, next it is treated by an enzyme called as decarboxylase enzyme. Friends, understand what the transaminase enzyme does. Transaminase enzyme remove the amino group. What the carboxylase enzyme will do? Remove the carboxyl group. Simple. So from the IPA there is a removal of the carboxyl group. Upon removal of the carboxyl group, we are getting indole 3 acetaldehyde. What we are getting? Indole 3 acetaldehyde. Very simple. Now this indole 3 acetaldehyde treated by an enzyme called as dehydrogenase enzyme. Upon treating with dehydrogenase enzyme, ultimately what do we got? We got the indole acetic acid. Such a simple pathway it is. No need to take any tension or stress. Easily you can remember. What's the first point? Tryptophan. Tryptophan can be treated by which enzyme? Tryptophan transaminase. Transfer the amino group. Because of the transfer of amino group, tryptophan converted into indole 3-pyruvic acid. Now the indole 3-pyruvic acid, upon removal of the carboxyl group by a decarboxylase enzyme, converted into indole 3 acetaldehyde. Now that indole 3 acetaldehyde treated by a dehydrogenase enzyme, giving rise to what, friends? Indole acetic acid. Such a simple pathway. You people no need to remember the structure. If you are remembering the structure unnecessarily, you are literally wasting the time. Okay, so I want you people majorly remember the structure, only this. Which one? The tryptophan structure. Apart from that, you don't need to remember anything at all. Okay, so friends, now we have clearly studied about this particular auxin biosynthesis. The last and final concept before ending the class we have to study is the transport of auxin. If you look into the 2018 question paper and if you look into the 2017 question paper also, in both times we got the question about the transport of auxin. So let me tell you, I will prove what we are studying. We were studying that auxin will be synthesized in the shoot and auxin will be traveling to the root. That is the reason we are calling it as the auxin as a rooting hormone. Yes or no, friends? Of course, yes. Now let me prove to all of you. Polar auxin transport. Let me explain you. All the students look into the diagram. Such a simple and classical experiment. So this is called coleoptile. Understand, friends? See, middle part of the coleoptile, what do you call it as? You call it as hypocotyl. And upper part of the coleoptile, what do you call it as? You call it as the epicotyl. Middle part is called as hypocotyl, upper part is called as epicotyl. What I have done? I have taken the hypocotyl. I cut the hypocotyl. Follow me, every student, look at the diagram. I cut the hypocotyl. After cutting the hypocotyl, I kept this hypocotyl between the two agar blocks. So this is one agar block, this is agar block, and this is agar block. Between the two agar blocks, what did I kept? I kept the hypocotyl. Now every student follow my words very correctly. What is happening now? Upper agar block, you can see, right, it is a very dark blue color. Why? Because it is completely loaded with auxin. Follow my words correctly. Upper agar block completely loaded with auxin. And what about the lower agar block? It is completely white in color. It is not at all loaded with anything. After some time, what we observed, you all the students look into this, the dark blue color one became light blue color one. The complete white one also became a light blue color. What do you mean by that? What do you understand by that simple experiment? The auxin starts moving from the upper agar block to the lower agar block. So what do you mean by that? Auxin always move from shoot to root. That's a simple experiment. Now this time, every student listen, look, follow the diagram very correctly. This time, see, originally coleoptile is like this, yes or no? Coleoptile is like this. My upper part is the shoot and this part is called as root. You know very well. Now this time what I have done, I have inverted it. See, originally this is how the coleoptile looks like. Now I have inverted this, which means this became root, this became root, this became shoot. Now every student remember. So this became a root part and this became a shoot part. Correct na? Once again, we cut the middle part, that is called as hypocotyl. What I have done? Once again, I kept the hypocotyl between the agar blocks. Once again, upper agar block is loaded with auxin, lower agar block is not loaded with anything. After some time, you tell me, is there any change occur? No change. However, the dark blue color agar block will be same. However, the light white color agar block, that is also same. There will be no change. So what do you understand? You have to you have to understand the statement that auxin never ever, follow my words, auxin never ever move from root to shoot. It never moves. Auxin always moves where, friends? From shoot to the root. Where it will move? Shoot to the root. Friends, this kind of a unidirectional transport of auxin. What transport? Unidirectional, which is moving only in one direction, which we can call the polar auxin transport. You can also call it as basipetal transport from the shoot tip towards the root. Right? This is called polar auxin transport. Okay. So friends, that's all for now. I hope every one of you understand the concept very correctly. Now the continuation of this will occur in the app 11:30 AM. Okay. So whomever paid, I have already enrolled everyone. So you automatically get the link in the 11:30 AM. If you just click the link, automatically you can enter into the classroom. Okay. So in the 11:30 class, I will finish the auxin and I will do my best to finish even gibberellic acid also. So as a result, 7 PM class, we will do the revision of both hormones thoroughly. Okay. So for the revision class, just keep the PPT with you. PPT I will upload in the app. Just keep the PPT with you and keep the Pathfinder with you. I will tell you how to study exactly. Okay. I hope it's clear for everyone. If you are having any kind of a doubt, even as a small doubt or big doubt, please ask me in the morning 11:30 AM class. Okay. I hope the discussion is useful, and I am requesting every one of you be consistent your preparation till January 20 or whatever it may be. Okay. So please be consistent throughout the preparation. Okay. Okay, friends. Thank you all. Thank you so much. Thank you for your patience. Thank you all.